Welcome. In today's lecture, we'll be going over Chapter 1. Now, before we begin, there's a couple of things that I would like to go over. There is a lot of information that you're going to need to know in every chapter as we move through the semester. So, to help with this process, we have provided a downloadable copy of each of the PowerPoint presentations that will appear in these videos. Take a moment, pause this video, go download that PowerPoint file, and come back and be prepared to take notes as we move from slide to slide. So let's jump right in with anatomy. Anatomy and physiology are all about human structure and function. We are studying the biology of the human body, looking at how your body works. And this is extremely important and will be critical for your future careers, whether you're going into nursing, rad tech, dental hygiene, exercise physiology, pathophysiology, or medicine itself. Anatomy and physiology is absolutely foundational for each of your careers. As we move through the semester, you're going to see a series of slides that say expected learning outcomes. The goal for these slides is not only to give you a guide of what's coming ahead, but for you to come back to these slides and assess your own learning. So after reading the text and watching this video, I'd like you to come back to this slide and ask yourself, without looking at your notes, can you define anatomy and physiology? Can you describe several ways of studying human anatomy? Can you define a few subdisciplines of human physiology? Let's start with the difference between anatomy and physiology. Anatomy is examining the structure of the human body. Physiology is the study of its function. Anatomy and physiology complement each other, and they can't be entirely separated. There is unity between form and function. If we look at an example, if we look at the anatomy of the heart, we're looking at the vessels and the muscles, all of the structures in the heart, like the aorta. However, if we want to look at the physiology, we could look at something like an ECG, looking at the rhythms of the heart, how it functions. Those are the differences between those two. Now, there are many different ways that we can study anatomy. Early scientists use exploratory surgery by opening up the human body and seeing what they find. However, nowadays we have an advantage. We have medical imaging, so we are able to view inside the body without doing surgery or putting anyone at risk. And there's an entire branch of study that is dedicated to medical imaging, and that is called radiology. If we want to study structures that can be seen with the naked eye, we call that gross anatomy. However, if we want to study something that's microscopic, we call that histology. Histopathology would be the microscopic examination of tissues for signs of disease. And cytology is the study of the structure and function of cells themselves. Lastly, ultrastructure is where we have an electron microscope where we can view very specific details that we cannot see in any other form. Physiology has many subdisciplines, and we have a few examples here that we're going to go through. Neurophysiology. Anytime we hear neuro, we're referring to the nervous system, and physiology is for physiology. So neurophysiology is the physiology of the nervous system. Endocrinology is the physiology of hormones. And pathophysiology is looking at the mechanisms of disease. We are not always fortunate enough to do all of our studies on human bodies. And so we have what is called comparative physiology, the study of different species to learn about body functions. I'm sure many of you have heard of scientific research using rodents or some other type of animal for scientific investigation. Our next section is looking at the scientific method. We'll be starting with the inductive method and the hypothetical deductive method and noting the differences between the two. The inductive method, where we make numerous observations until we become confident in drawing generalizations and predictions. This is our method for gaining knowledge of anatomy. But what does that mean? What it means is, if I take this clicker here and I had no idea what's going to happen with it, if I let it go, where is it going? Is it going up to the ceiling or is it going down to the ground? Well, I can repeat this over and over and over again, and consistently every time the clicker will drop to the ground. I have proof now that this is going to work. Proof in science means that you've made reliable observations that have been repeatedly confirmed and have not been falsified by any credible observation. Just remember that all truth in science is tentative. It's proof beyond a reasonable doubt. Our hypothetico-deductive method is how we gain physiological knowledge. In this case, we have an investigator that formulates a hypothesis. an educated guess, if you will, or a possible answer to a question. The investigator has to form a good hypothesis that has to be consistent with what is already known and testable. That means is I can't walk into a lab and say, I have a great hypothesis. If I let this clicker go, it's going up to the ceiling. I'll be laughed out of the lab. Everyone knows that there has been consistent results that say the clicker will go to the ground. So I have to keep that in mind and use my pool of knowledge to make a good hypothesis. Now, if we go through this process and we want to claim that something is scientifically true, we must be able to specify what evidence it would take to prove it wrong. If we cannot prove something wrong, then it is not scientific fact. Now, when we're moving into the research phase, we need to look at our experimental design. And there's a couple of factors that we need to keep in mind. First and foremost will be our sample size. That's going to be the number of subjects in our study. If I'm doing research on human behavior and I only choose 2 subjects to study from, will I be able to make generalizations about the human population? Absolutely not. However, if I up my sample size to 20,000, then I can start to see patterns of human behavior. When also doing test studies, for example, drug test studies, we want to look at what's called controls. Usually in a test study, we're going to have 2 groups, the treatment group and the control group. The difference between these 2 is vital for our study. The treatment group, for example, if we were doing a cancer study, would be receiving a pill with the cancer drug. Our control group would be receiving what is called a placebo. It appears to be the cancer drug, but it doesn't have any of the cancer drug inside of it. Why this is done is because of psychosomatic effects, the effects of the subject's state of mind on his or her physiology. So if we told the test group that they had the treatment and we told the control group that they had the placebo, their state of mind can affect their physiology. And we want to make sure that we have consistent and reliable results. So we make sure that we have a blind study. None of the patients know which group they are in. However, what happens if the experimenter knows which pills are the placebo and which pills are the treatment? Let's say I'm the experimenter and I was driving to work this morning trying to get there so I could start the drug study. One of the patients cuts me off in traffic. Do you think I'm going to give them the placebo or the treatment? There's a good chance that I could give them the placebo. Now, that's not going to happen in every case, but just to avoid that, we do what is called a double-blind study. Neither the patient nor the experimenter knows which pill is the placebo and which pill is the medicine. Lastly, we have to have statistical testing. It has to provide a statement of probability that the treatment was effective. Once we're done with our study, we have to move into the phase of peer review. This is where our study goes under critical evaluation by other experts in the field. So peer review would not be considered efficient if your mother were to look at your scientific study and say, job well done. It needs to be done by other knowledgeable people within that field. This needs to be done before funding, before publication, and done by using verification and repeatability of results. This will ensure honesty, objectivity, and quality in science, which we all value. Some terminology that we need to know. If we call something scientific fact, that information can be independently verified. That means if I run the experiment here in Chattanooga, Tennessee, someone else in Washington or Russia or London or Brazil can do the same study and come up with the same results. If we call something a law of nature, that is a generalization about the way matter and energy behave. And these results come from inductive reasoning and repeated observations. If we call something a theory, that is an explanatory statement or set of statements derived from facts, laws, and confirmed hypotheses. And there's many of these theories that we're going to look at as we move through the semester, like the cell theory, the fluid mosaic theory, or the sliding filament theory when we talk about muscle contraction. Our next section is looking at human structure and its complexities. The human body is very complex, and there is a hierarchy to that complexity. The human body as a whole is considered an organism. This organism is made up of organ systems. That be the digestive system, the nervous system, the skeletal system, the muscular system. There's a whole host of systems that come together to form the organism. But each organ system is composed of organs. Organs are composed of tissues. Tissues are composed of different types of cells. Cells are composed of organelles. Now that might seem like a strange word, but I'm sure we've heard of an organelle in our lives. Does anyone remember what the powerhouse of the cell is? Of course, it's mitochondria. That is an organelle. And each of these organelles are composed of molecules, and the molecules are composed of atoms. Atoms we'll discuss more in depth in chapter 2. So let's look at some examples. If we look at the baby here, this is our organism. It's made up of organ systems, and our example here is the digestive system. One of the main organs in the digestive system is the stomach. The stomach is composed of tissues that are composed of cells, that are composed of organelles like that mitochondria, or the nucleus, for example, that houses the molecules called DNA. And then we have, lastly, our atoms. Now, we've looked a lot at anatomy. We know what it is. We know it's different from physiology. But I want to make note that no two humans are exactly alike. They have anatomical variation. So when we look at our textbook, it's showing the most common organizations of structures. Just keep in mind that some individuals will lack certain muscles, some will have atypical number of vertebrae, some individuals have atypical number of certain organs, and some will even have what's called situs inversus, where you have an organ that we expect to be on one side of the body, however we find the organ placement on another. Let's look at some examples. Our first example is going to be the normal orientation of the kidneys. We typically find one on the right and one on the left, and they're about the same height. However, we can have an abnormal orientation where we have one that's taller than the other. Or we can have a horseshoe kidney, where they're connected like a horseshoe to one another. We can also see anatomical variation in other areas, like the aorta. We have the branching of the aorta that is typical, but we can also see that there are many examples where that branching can be slightly different. When we move into A&P2, and we're studying the cardiovascular system, we will learn the most common orientation for branching of the aorta. In our next section, we're going to start with looking at the characteristics of life and their importance. All of these characteristics that we're going to see on the next 2 slides have to be seen in order for something to be considered living. If you only have one, it's not considered a living being. We need the collective set of characteristics to show life. Our first characteristic is organization. Living things exhibit a much higher level of organization than non-living things. Cellular composition, like our cell theory, that all living things are made up of cells. They're compartmentalized either into one cell, because we have unicellular organisms, or more cells. We have multicellular organisms. All living things have internal chemical reactions, and we call the collective metabolism. Responsiveness is another characteristic. That's the ability to sense and react to stimuli. Living things must have movement, either of the organism itself or substances within the organism. Next, each living thing has to maintain a relatively stable internal condition. That is called homeostasis. Development. We must see differentiation and growth. Reproduction. Each living thing must be able to produce copies of itself. And lastly, evolution of a population. We must have the ability to see a change in genes. Now we've talked about anatomical variation, we're going to look at physiological variation. And this depends on sex, age, weight, physical activity, even the environment. So when we look at physiological values in our textbook, we're basing those off of a reference man and a reference woman. So the details for our reference man are going to be 22 years old, about 154 pounds with light physical activity. Now, his physiological values will be different than a 65-year-old man who's 284 pounds that does no physical activity. So failure to consider these variations can lead to over-medication of the elderly or medicating women on the basis of research done on men. So we have to always remember that physiological variation exists and each patient must be treated individually. Next, we're going to look at homeostasis in detail. We said that homeostasis is one of the characteristics of life. However, let's expound upon that. Homeostasis is the ability to detect change, activate mechanisms to oppose it, and then maintain a relatively stable internal environment. Negative feedback is what allows for dynamic equilibrium, keeping the body in homeostasis. And this is fundamental for the body, because if we lose homeostatic control, this can lead to illness or even death. So because feedback mechanisms alter the original changes that triggered them, they're called feedback loops. And one of the main examples we can use for homeostasis is body temperature. If your body gets too warm, the vessels in your skin are going to dilate and sweating will begin. This will allow the body to cool down. However, when your body gets too cold, those same vessels will constrict and shivering will begin, and the body will warm back up. So let's look at this in terms of a thermostat. Say you've set the temperature in your house to 68 degrees. When the room temperature falls to 67, the thermostat is going to turn on the furnace. The furnace is going to allow a heat output. The room temperature is going to rise back up towards that 68 degrees. Once it reaches 68 degrees, the thermostat will signal the furnace to shut off, and the room will begin to cool again. And we will continue on this cycle until the AC is turned off, or the heat is turned off. That is our feedback loop. This is exactly what's happening in the body as we get warm and cool and warm and cool. As the body gets too cold, we have vasoconstriction until eventually that shivering begins, that muscle movement causing the body to warm up. And then when the body gets too hot, we have that vasodilation, and then sweating will begin, that liquid cooling the body down. So when we're looking at homeostasis and negative feedback, there's a couple of terms that we need to know. The first being receptors. Those are the structures that sense change in the body. Whether that be a chemical change, a temperature change, a pressure change, receptors are going to sense that change. We have our integrating or control center that processes the sensory information and makes a decision, and then directs a response. And lastly, we have what is called an effector. It's the cellular organ that carries out the final corrective action to restore homeostasis. Positive feedback, however, is how we get rapid change. It is a self-amplifying cycle. It leads to greater change in the same direction. So our feedback loop is repeated. It is how we get rapid change that we see in childbirth or blood clotting or protein digestion. But it can also sometimes be very dangerous, where you have a vicious cycle of a runaway fever. So let's look at one of those processes of rapid change, childbirth. We can see here in our image that we have a fetus that's ready to come out. It's pushing against the cervix. There's a nerve signal that's going up to the brain saying that there's pressure on the cervix. That causes the pituitary gland to release oxytocin, and then that will cause uterine contractions, which then further pushes the fetus onto the cervix, continuing this feedback until we have the end result, which is the birth of the child. There's a couple of terms that we need to talk about when it comes to gradients and flow. Gradients is a difference in chemical concentration, charge, temperature, or pressure between 2 points. Matter and energy tend to flow down these gradients. For example, your blood flows from a place of higher pressure to a place of lower pressure. If we have any movement in the opposite direction, we call that going up the gradient, and this requires the spending of metabolic energy. There are different kinds of gradients. We have chemicals that flow down concentration gradients, charged particles that flow down electrical gradients, and heat flows down thermal gradients. So let's look at this visually with the example of the boy on the wagon. Sitting at the top of the hill, the boy sits in the wagon and he rolls down the hill. There is no energy that's required. Gravity will do the work. The boy will slide effortlessly down the hill. That represents going down a gradient, whether that be down a concentration gradient. However, when the boy reaches the bottom of the hill, he needs to turn around and lug that wagon all the way up to the top. That takes more energy than going down. That represents doing work. And so that represents going up a concentration gradient. So we have a couple examples of how we're going to see this come into play in anatomy and physiology. Let's look at our first example. We have glucose here. There's a large number of glucose on one location and a smaller number of glucose in the other. Which direction do you think glucose is going to flow? Of course, it's going to flow from the area of higher concentration to the area of lower concentration. Lastly, we're going to look at the language of medicine. It is extremely important to be precise, especially when spelling terms, because there are many scientific terms that are spelled very similarly but have extremely different meanings. So in the healthcare professions, there is a demand for precision in order to maintain patient safety. And so we also want to hold that same standard as we start our careers in anatomy and physiology. So as we move through the semester, especially when going through the laboratory, you will see that spelling counts. So if there are some terms along the way that you will have difficulty spelling, I suggest that you take a note, write down a list of all the vocabulary words for that unit that are difficult for you to spell, and practice them before we reach the exam, and you are required to know their spelling. All right, so let's review what we've gone over so far in this chapter. We looked at the differences between anatomy and physiology. We talked about the characteristics of life, looking at the cell theory, all structures have activity of cells. Evolution, we have to see a change in genes. We talked about the hierarchy of complexity. The organism is the highest level all the way down to the smallest atoms. We talked about homeostasis, maintaining that internal stable condition. And we also talked about gradients and flow, looking at how matter and energy behave by flowing down gradients. This is at the foundation for where we're going in our next video, looking at Chapter 2. See you then.